Thank you very much for the invitation, and congratulations, and Soha, Martin, Elias, and everyone involved in the organization of this great meeting. And today I will talk about the transfer RNA and its participation in the origin of the biological system. So before I talk directly about the tRNAs, I need to say how as I define life, because this is a very important point, because establish a transitional point between the abiotic world to biotic world. So here in this lecture, I will define life as a code system, and you have a lot of examples of organic codes in the biological system. For example, the genetic code, regulation of gene expression is by code, hormone receptor signalization is also by a code, histone code, epigenetic code, and there are a lot of other codes in the biological system. So we can define a living being as multiple layers of the codes, and I need define a code or say the characteristics that you need to identify a code, and a code must link two words, one being defined as a signal and the other as a biological meaning. But the connection between the two things, I need an adapter molecule which establish the rules of correlation and make contact between the signal and its meaning. It's a very important thing. So with this, from this point, the next question is, what was the first code? This is important because from my concept of life, if I can identify the first code, I identify the exact point, the exactly transitional point between the abiotic world to biotic world. And the answer to this question is the genetic code. The genetic code is a very important code, and the genetic code establishes the connection between the nucleic acids and protein, and I'm sure that was the first code. So when you talk about the genetic code, we need to also talk about the decoding system. In this case, the translation system. And in this point, I will start to talk about the tRNAs. There are a lot of evidence or suggestions of the participation of the tRNAs in the initial organization of the biological systems. Here we have, in this picture, you have the tRNAs and the secondary structure and the three-dimensional structure. And here I will highlight two portions, very important portion here. And the acceptor arm or acceptor stem. In this point, the amino acid tRNA synthetase bind an amino acid, and this part of the tRNA interacted directly with the peptidyltransferase center in the large subunit of the ribosome. In the other side, we have the anticodon loop that interacted with the small subunit, and more specifically with the mRNA that are binding in the small subunit in the decode system site. So the question here is, what the correlation between the tRNAs and the ribosome? And I will show first data about the large subunit. After this, I will show data about the small subunit. To try and understand the correlation between the tRNAs and the ribosomal RNAs, more specifically the 23S RNA, we reconstructed the ancestral sequence for each one kind of the tRNAs. After this, we compare this ancestral sequence with the catalytic site of the ribosome. And in this table, you can see the result of this analysis, this initial analysis. Here we have the cognate tRNA ancestral sequence, whole kind. And here we have the initial position and the final position that this tRNA had matched with the catalytic site in the ribosome. And here in the last, here we have the percentage of the similarity between them. For my surprise, a very interesting result. We found a very interesting result. For example, here, for ancestral to leucine tRNA, we had initial match in the position 54 in the catalytic site of the ribosome, and the final match in the position 92. When you look for the serine, for example, serine had initial match in the position 127, and the final match in the position 151. And if you look at other example, histidine have initial match in the position 164, and the final match in the position 220. And as you see, in the ribosome, you had a match with different kinds of tRNAs in different points, but like a side by side, like a fingerprint. We can find a fingerprint of the ancestral tRNAs inside of the ribosome, mainly inside of the catalytic site of the ribosome. And similarity is side by side. And from this data, we constructed a concatemer and compare this concatemer with the catalytic site of the ribosome. And here we can see the alignment. We have a similar around 52%. It's a good similarity to primary sequence. And a very important point here is this adenine are conserved in the ancestral sequence. And this adenine is very important to the catalysis. From this ancestral sequence that we constructed based in the tRNA ancestral sequence, we reconstructed the three-dimensional structure. And you can see here in red, this is the structure derived from the primary sequence that we reconstructed based on the tRNA ancestral sequence. In blue, we have the modern catalytic site of the ribosome. When you did the structural alignment, the similarity between the ancestral structure in red and the modern structure in blue was 92%, very high similarity. And after this, we did a molecular docking between the tRNAs and the ancestral structure, and the results show that in all the case, the tRNAs interacted with the catalytic site, ancestral catalytic site of the ribosome. And in all cases, this interaction is by the acceptor arm, like we can see in the cells or in the modern process of translation. And other important point here is that in all cases, the anticodon are free to establish other connection. For example, with other RNA molecule like mRNA or primitive mRNA. So with this, we had some evidence that the catalytic sites of the ribosome was formed by junctions of the small RNAs, and these small RNAs was derived from the tRNA's ancestors. We did the same analysis to the small subunit, and it was possible constructed at the same way and concatenate and compare with the 16S RNA, ribosomal RNA, and again we have a similarity around 51–52%. From this ancestral sequence, we constructed a three-dimensional structure and compare with the modern structures to the 16S ribosomal sequence, and we have again a high similarity between the ancestral sequence, ancestral structure, and the modern structure. Here you can see the similarity with the ribosomal RNA to Thermophilus was around 94%, E. coli around 9%, and Mycobacterium magmatis around 86%. It's very high. So with this data, from this data, we reconstructed the primitive translation system, right? Here in green, we have the large subunity or the primitive large subunity of the ribosome. In blue, we have the tRNAs, and in orange, an mRNA, and in red, our ancestral structure. It's very interesting because this ancestral structure is very similar to the code system when the mRNA binds during the translation process. So we had awesome evidence that the small subunit was formed by junction of the ancestral tRNAs, like the large subunit of the 23S RNA molecule. So the model that we constructed to explain the evolution and the origin of the primitive translation system, and we have a tRNA here and some kinds of tRNA by junction gave origin to the peptidyl center, that is the catalytic site of the ribosome, and gave origin to the cold site in the small subunit of the ribosome, in this case, the primitive ribosome. And the PTC could establish an interaction with the tRNAs, and the cold site in the other side here could establish an interaction with the other RNA, in this case the mRNA. The interaction between these two complexes, the PTC with the tRNAs and then the cold site with the mRNA, gave origin to the primitive translation system. Okay, so until now I talk and I show some evidence about the correlation between the ancestral tRNAs and the formation of the ribosomal molecules, the catalytic site in the large subunit and the decode site in the small subunit. But I don't talk nothing about this third element, that is the mRNA. Okay, so I will pass a little fast about this data, but we did an analysis when we took our ancestral sequence to each one tRNA and did a combination 3 by 3 and compare with the modern genes. When we did this, we found a very interesting result, because we found match between the ancestral sequence of the tRNAs with the basic pathways in modern genes. So we published this paper in 2016, when we proposed a proteome before the last universal common ancestor. We found similarities with many enzymes that worked in the glycolysis. And a very important point here is that in all cases that I found similarity between the tRNA ancestors and the modern genes to glycolysis, the similarity was in the catalytic site. Here in red, and you can see the structural reconstruction based on the tRNA ancestral sequence. And we also found similarity between the ancestral tRNA sequence and a very important enzyme, the RNA-dependent RNA polymerase. This is very important because this enzyme replicates RNA in other RNA molecule. And similar what we found to the glycolysis, the part of the gene that had similarity with the ancestral sequence to tRNA is the catalytic site. When you compare the structure that you constructed based in the tRNA ancestral sequence with the modern protein, we had a very high structural similarity, mainly in the palm domain, that is the catalytic domain of the polymerases. And here we can see a practically identical structure between the ancestral genes and the modern genes to the same protein. And we took these structures, the ancestral structures and the modern structures, to the RNA-dependent RNA polymerase and did it tree of distance. It's not a phylogenetic analysis. It's just an analysis of the structural distance between the structures. But we found a very interesting result here. And we can see here the small structure, this small structure here, are derived from the ancestral tRNA sequence. And here we have the modern structures that you can find in many groups of virus. And in this tree, we can see that the ancestral sequence had more similarity with the ancient groups of virus. And now there are a lot of discussion about the origin of life in the same time that the initial organization of the biological system. I don't know if Gustavo will talk about this in your lecture, but a lot of researchers I talk about this with a lot of evidence of this ancient orange to the virus. And this was a very interesting result because the structural similarity between the tRNA ancestral sequence is with the ancient virus group. So I think that my time is gone. Here I simplified, summarize my model, and in the beginnings of the origin of the biological system or life, we had the tRNAs that, by a process of junction of these tRNAs, gave you orange to the mRNAs or the first genes, and gave you also orange to the catalytic sites of the ribosome and the decode site of the ribosome. With this was possible the emergence of the primitive translation system, and the data that I show you here make it possible to suggest that the tRNAs orchestrated the initial organization of life. In this case, I suggest here that the tRNA worked as a demiurge in the origin of life. And I would like to thank my collaborators, and thank you very much. I'm open to questions.